Well, welcome to A Penny for Your Thoughts. We are live here on Iowa State University campus. Get to interview some really good friends, mentors of mine, people that have impacted my career in an immense way. It's not often you get to be in these positions where you get to thank the people that have helped you get to where you're at. And I have Alison and Robertson and Daren Mueller here. Again, it's not often you get to be in these positions. So, you know, with our audience across the U.S. and now I guess what I learned globally the other day from you, I just wanted to thank them. You know, I often make the joke, and when I'm talking to other people, I guess I've never really told you guys this, but I often see you guys as somewhat like not parent figures, but the way you handled all my annoying questions. It reminded me of like a toddler and a parent. You know, I just remember asking you guys so many questions, and you never got annoyed with me. You always fostered my questions, and I think that really put me in a position to always want to continue to learn and obviously learn some. So
that's the reason you tolerate me?
Yeah,
that you're paying them back for.
Yeah.
Is he the toddler and you're the parent now?
Yeah, that's scary.
Learn from the best. So no, it's pretty cool to be able to interview them. You know, obviously 2025 was a really challenging year. Maybe we'll say an exciting year for pathologists. You know, with all these new diseases that we're talking about in the Corn Belt. So looking forward to picking their brain. You know, it's going to be a little bit different than our normal episodes. You know, we're still going to stick with the science and management portion. So we'll have part 1 and part 2. But we're going to, you know, instead of focusing on one topic, we're going to kind of scatter across a range of different diseases. So I hold these guys in pretty high standards. But we're really going to put them on the spot and test them because we're going to go from disease to disease and test their knowledge on the spot as opposed to knowing and focusing on one disease. So I'm looking forward to the conversation.
I love it. Well, let's start with, I guess, thank you guys for letting us be here on campus with you today. And it was fun. Allison actually walked in the room and said, well, this is intimidating. So for those of you that can't see our setup, we now have lights and cameras, and it's a lot of fun. But let's just start. Allison, I'll have you go first, and then Daren. Most of our listeners have heard episodes with you guys before, but just give us a quick recap who you are and the work you're doing here at Iowa State.
First of all, I would like to say this is the thanks we get.
Exactly.
Put you on the spot, making you nervous, making you sweat potentially.
Yeah.
So I'm Alison Robertson. I'm a professor in field crops extension pathologist here at Iowa State. I started in 2004. I work predominantly with corn, a little bit with soybeans.
And I have the exact same answer, except I'm predominantly soybeans and then a little bit in corn. And then I also get to put the hat on of the integrated pest management coordinator. So a little bit of semi-administrative stuff.
So before Sean goes into his other question, I wanted to give you guys a little bit of time to highlight the research your labs do. You know, me coming from your labs, doing work with y'all. It's pretty cool. I mean, obviously we're in one of the I states doing research on corn and soybeans that drives every conversation we have. Allison, can you tell me about some of the students, where they're from, some of the work you're doing currently?
Sure. So I think one of the things that I like being an extension for is that I get to talk with farmers a lot. And so a lot of my research is driven by the questions that those farmers have. So at the moment in my lab, I have a student who's working on cover crops. We're trying to understand cereal rye ahead of corn and why we sometimes get those yield declines. I have that student's from Costa Rica. I have another student from Kenya who's working on crown rot on corn, which has been a bit of an issue the last few years.
Maybe the understatement of the day.
I have another student from Zimbabwe who is working on understanding foliar disease management in corn. So at the moment he's looking at timing and planting date, and then timing of fungicide application. What is best to get the highest yield, right? And then I have a fourth student who's working on Phytophthora soji of soybean, but from the molecular angle. So he's doing a lot of bioinformatics and stuff like that. So we work with another professor at Arkansas State University because obviously I don't have a lot of expertise in that area.
Yeah,
so thank
molecular
goodness
is
for a
another level.
seller. Yeah.
Darren, what about you?
Yeah, I don't have the molecular part of the lab at all. So right now I have, we mostly focus on soybean diseases. SDS is probably the predominant one, although lately we've been really trying to revisit foliar diseases and then also seed diseases. And so I have one student that's looking at interactions between planting date and maturity groups and then what diseases show up. And he's from Costa Rica. I have another one that's looking at, we're really taking a fresh look at the foliar spots. And so we did this last year, and this is like sort of admitting, like, you know, we all continue to learn, but like we have a fungicide resistance is a big issue, and brown spot is one of the diseases that we sort of use as the canary in the mine. And, and so I sent the lab out, and we were going to collect all the brown spot, you know, leaves with brown spot on it. And it's for, I mean, most of us know what that looks like, and it's pretty easy. We sent out a crew, several crews. We all came back, and 90% of them weren't brown spot.
Really?
So yeah, including a bunch of the leaves I collected. You know, and I was like, oh crap. So
Nailed it.
And so you know, a lot of them ended up being a different disease, and so we're starting to unpack what we're seeing and why we're seeing it, and sort of making sure that what recommendations we're making are accurate. So let's see, we have I have a postdoc working on SDS and sort of what are the conditions that sort of increase the risk. PhD student that's looking at developing a predictive model for frog eye leaf spot and sort of when and why it shows up, and sort of the weather conditions that would sort of drive that. And so my students are 3 from Costa Rica, and then I have 1 from Nigeria. My 2 American students actually just graduated, and they're off in industry now. So, and then 2 postdocs, 1 from Mexico, 1 from Nepal. So
Nice. Well, I look forward to talking about some of these diseases.
No, that's good. Well, let's go ahead and dive in. And you guys have both been guests on our show before, but one of our favorite things to ask, you know, you guys are doing a lot of hard work in the industry. You're also really in tune with what's happening in the field. But we like to start with just kind of asking the question, is there anything kind of new or exciting that has you excited about agriculture today? Just whether it's something you've seen from your students or something out of industry, doesn't have to be directly related to the work that you're doing.
you want me to start?
Yes, please.
Okay, yeah, I would say like for me it's sort of a corny answer, but it's the drones.
Yeah.
And so it's, you know, like I think this is a great year as an example as to why it's important to continue to embrace new technology and try to figure it out. And you know, are we using it as best as we can? Probably not. We still have a lot of things we need to learn about. But you know, in Iowa this year we had 30, 40 days in a row where it was too wet to get into the field, at least, you know, safely, and you know, and airplanes can't handle all of the acres, and so it was great to have so many drones out there spraying, and you know, and we have a lot to learn and a lot to go, but that's just like a good example of like, you know, agriculture in general, and you know, looking at embracing, adopting, and sort of using the new technologies, and you know, it's never ending, so it's always exciting.
Yeah.
From the original, you know, you think about the innovation that started when we figured out how to first turn the earth. I haven't been negative about drones, but in my mind, I'm like, man, we have so many great tools already. Why are we putting so much effort into this? And then to your point, there's a lot of fields that would not have gotten an application if it wasn't for the amount of drones in the marketplace. So there is still a ton to learn, but without that tool, we would have been in a pretty tough spot for sure.
Yeah.
So I was thinking about this question when Andrew sent it to me last night. And I think both of you know that my middle name is Skeptical. And so
That's actually not true. I know what it is.
And so I think there's a lot of new technology out there, including the drones, but I'm thinking also of kind of the biological technology that's out there. And then the other thing that I've been thinking a lot about is this detecting pathogen technology that's out there, right? And I know that Daren and I specifically talked about the biologicals, that it's very hard to show benefit for them. That's what I find in my research. And so we were talking about this the other day, is what are we missing, right? What are we not measuring that maybe we should be? And it's the same with like the pathogen detection, right? You guys also know that I'm totally obsessed with the disease triangle. And so just the fact that you detected the pathogen does not mean you're going to have the disease, right? And so I was thinking about your question because I was like, yeah, there's a number of great technologies that are coming forward in agriculture. But I'm still so skeptical about them. The same with AI, right? If you think about AI, but there's so much rubbish going into AI that there's a lot of rubbish coming out. And so
Rubbish in, rubbish out.
Word of the day, rubbish.
Why? What would you say?
That's a beautiful. No, it's a beautiful word. I love it.
That's what I would use. So
completely
we don't want Sean to have to use the beeper to blur you out or to beep you out.
So yeah. So I mean, a lot of great technologies, but I'm skeptical. Maybe it's because I know too much or too little, or I'm just not measuring the right stuff.
It is tricky. I mean, I'm excited too about all the technology to detect diseases earlier than what we've traditionally have in the past. And you start looking at some of these biologicals, it's tricky because you got Mother Nature that impacts all that stuff, and it is, you either know too much or know too little, right?
Yep.
So.
Yeah, specifically in that range too, it's hard because commercially we want a product to kind of fit broad acres, right?
Mm-hmm.
It's hard to be as granularly prescriptive as we probably need to be.
Mm-hmm.
So then I think sometimes we rule things out because we shotgun them into the field.
Yeah.
Yes.
And they probably do work or have some impact. We're probably just not necessarily using them in the right way. And so there's a lot of work to be done there. But let's dive into our conversation, Andrew. So I thought you did a really nice job just setting up kind of the goal of today. But I think really what we kind of want to do is maybe just spend some time talking about your key takeaways from 2025 and then auguring into some specific diseases and having you guys hopefully help us understand some of those a little bit. So maybe, Daren, if you want to go first, just give us your key observations of 2025, and then we'll go to you, Alison.
Yeah, I would say I'm going to answer it in the corn world, and our introductions sort of were, she handles corn, I handle soybeans, but, corn diseases probably were the first and foremost, thing in people's conversations. And I would say, whenever you have a year like this, a lot of times, like by the time, NBC and all these other people that usually don't call us, were calling us and asking us what was going on, that we sort of had the attention of a little bit broader audience. And so, like, what happened this year compared to other years? And I think one of the coolest things is, like, we go back, we actually can track disease, and Alison pointed this out at one of our field days, was, Southern rust showed up about the exact same time in 2024 as in 2025. So why did we have 2024 where we sort of like I'm going to call it more of a normal year, and then 2025 is like sort of a worst case scenario for farmers? Yeah, right. The differences are crazy. And so, and Alison is obsessed with the disease triangle, but though the spores landed in the perfect environment, and so, Sean, I know you're a golfer, but like, if you sneak out and go golfing at 6 a.m. to try to get a quick 9 in, like, you're sweating through your shirt by the second hole. And like it's in like I usually go out and for runs in the morning or whatever, and it's like I'll come back and I'm like, I'm too gross to go in the house, and like, I'll be ran a mile, and so like having that's just so much humidity and the warmer weather, like it was the perfect environment for southern rust this year, and then we had it for 5 weeks straight or whatever it was, and so. 2024, the spores were here, the inoculum was here, the same hybrids that were somewhat susceptible were here, but it's just a completely different story. And just being able to sort of reflect on that, and maybe for 2026, what lessons did we learn? Do we need to hit the panic button? Do we need to permanently change production practices? What are we looking at? So that's sort of my big takeaway.
Yeah.
My takeaway is that, yes, I mean, we had historic levels of southern rust this year. We had tar spot detected pretty early during the growing season, and once again, everybody was worried about tar spot. But I think with the southern rust coming in, there was just no real estate on the leaf for the tar spot. So yes, we've seen tar spot coming in at the end of the growing season, but yeah, southern rust like I've never seen before. A really cool year for a plant pathologist, but yeah, not really for a farmer.
One of my favorite pictures of the year was Megan Anderson posted on her X account, and she looked like she gets her spray tans from the same guy that Donald Trump does. You know, I mean, she was just rust red. Her entire body was. After
That reminds me of that combine video too.
Oh yeah,
where that combine was going through the corn.
Yeah,
and it was just
clouds.
Yeah, it was just
Yeah.
orange, yellowish orange. No, I mean that it is. What a weird year. You know, I remember last year too. I feel like that was some of the worst southern rust I've ever seen last year. And you have all these conversations. You're like, it can't really get worse than that in Iowa and the I-states. You know, in 2025 hits, and man, it wasn't even comparable. So it's weird, and I suppose that's a good. I mean, we were going to start the conversation off with southern rust because that was kind of the elephant in the room, right?
Mm-hmm.
What's your thoughts on that? I mean, going back, and you kind of touched on this, Daren. What do we know, and we'll kind of build up to this. What do we know about the overwintering process, alternative hosts, where this comes from, and then again, you alluded to it. What's the difference now versus? I mean, this disease has been around for as long as I can ever remember, and yet why is it just showing up now?
Yeah. I mean, one of the for some reason I was going back in the literature and trying to look at sort of like when southern rust has been a problem, or like when it was first showing up in Iowa. And there's a really good article in 1947, a huge outbreak of southern rust in Wisconsin. And so it has happened before, like all the stars have aligned in the past. But like if you look at it, like even, I think it was Don White, who's the corn pathologist at Illinois when I was a student there. He wrote the compendium of corn diseases, like, you know, sort of in some of his summaries in the mid 80s or late 80s, he's like, oh yeah, southern rust isn't really a problem in the Midwest. You know, like sort of really dismissed it as like, you know, it's a southern problem. And so there's a reason. It is a tropical disease. Like, you know, like Sean doesn't like to sweat through his shirt by the second hole, and none of us like the weather that we sort of experienced, you know, in Iowa. But like, that's what it likes. It likes, you know, it's used to more of tropical conditions, and so that's where it's going to thrive, or that's where it's going to, you know, be the problem. It also it's all rust diseases. They don't overwinter in the Midwest, and so they're called obligate parasites, which means they need green tissue to survive. And so that's the best thing about Iowa is and pathogens is that you get to winter and there's nothing living, and so all of the rust pathogens sort of have a huge reset, and then, you know, they a lot of them have, you know, different life cycles and different strategies to get back into Iowa the next year. But for southern rust, it has to survive somewhere, Mexico, southern Texas, somewhere in the south, and sort of go through all of the steps it needs to go through to get to Iowa. And if that's, you know, builds up an inoculum and moves very methodically, like what we think it did this year, or is it building up and then like a nice big hurricane or big pretty strong storm sort of gives it a big push, or, you know, whatever the conditions are. But it has to do, there's a bunch of steps down in the south that have to happen, and then we can sort of keep track of that and guess as to when it will get here.
Yeah. Do we know? I mean, thinking about that again, going back to, you know, you mentioned 1947. What's different about the way it's moving now versus? I mean, why is it rarely an issue? You know, tracking the weather. You know, you hear a lot about tropical storms and hurricanes. Is there truth to that, or is it more of environmental conditions from South America to Arkansas to Iowa and the movement of that pathogen?
I don't think the movement of the pathogen has changed at all. It's still coming up on those southerly winds from the south. This year, well, last year we had Tropical Storm Beryl or Hurricane Beryl, whichever you want to call it, that we think brought the spores up to Iowa. We saw southern rust about 7 days after Tropical Storm Beryl had brought winds up into Iowa. This year, if you look at the winds during June, we had more winds from the south, and they were stronger than we've had in previous years. So that was historic. So that was one thing that helped the inoculum get up here. And then the other thing was that there was probably a lot more inoculum down in the south because, according to Travis Faskey, not a lot of farmers, or a lot of farmers down there decided not to spray. And so that meant that there was more southern rust and more inoculum getting up here.
That I have not heard yet.
Yeah, we were talking about that, and I mean, so several different people have discussed that, and it's interesting to think about that. It's probably moving in a similar cadence most years. You take sub $4 corn and poor weather conditions, they stop spraying.
Yep.
All those spores, as they're landing, moving north, and we're not doing anything to combat them. And then you get up here and we have the type of weather that Daren's talking about. And it obviously, so the inoculum isn't just what arrived here, right? It's how much of it got knocked down in the process. And I don't think anybody, I mean, I hadn't heard anybody talk about that until a few weeks ago. And it was all of a sudden like, well, hey, wait a second, if nobody in the Rust Belt is doing anything to knock that pressure down, of course, it theoretically is going to make that worse. So you've kind of touched on this, but I guess I'll aim this at either one of you, but talk a little bit about what environmental conditions are favorable for the movement and arrival here.
So the spores have to get up here from the south, right? So we have to have winds or a hurricane or whatever that bring the spores up to us here. And then once they land here, they have to infect those corn leaves, right? And so we need at least, those corn leaves have to be wet for at least 6 hours for those spores to infect and to colonize that leaf. And then the other thing is we need very warm temperatures. So I think the optimal temperature for southern rust, if I've got it right, is 77 to 85 degrees. So that's pretty warm, right? And if you look at this past year, we had our nighttime temperatures didn't get below 70, 75. So we had higher than normal nighttime temperatures plus very warm daytime temperatures. So we had the right temperature conditions. And then we had those rains like every 4 or 5 days for 6 weeks, right? Which is what we didn't see in 2024.
Yeah, absolutely.
So you mentioned, that was one of my questions. You mentioned leaf wetness. What's the impact on leaf age? You know, if we track planting and vegetative growth from Texas to Arkansas to Iowa, what do we know? You know, if this comes in early or later, does the developmental stage of the crops in Illinois and Iowa matter? So leaf age.
So
this is a long answer, by the way.
I don't, when it comes to southern rust, no, I don't think so. I don't think leaf age is important with southern rust. It is with like gray leaf spot, right? Those leaves have to be a certain age. With common rust, supposedly there's adult plant maturity, so once we get into those reproductive growth stages, those leaves aren't as susceptible to common rust anymore. I'm not sure that I buy into that anymore. But I'm not sure that there's an effect of leaf age on susceptibility with southern rust. Do you know?
Well, I was going to say, I thought the angle you're going to take is your planting date study and then looking at disease development.
Yeah. So,
which was, that's a long answer I thought you were going to go with. So,
I mean, I can talk about that. So we have a study where we're planting. We have 2 planting dates. So April the 15th here in Ames. We also have Northwest and Southeast, and then May the 13th in Ames. So about a month apart. And what I found interesting was that as we were assessing those trials, we went in there 4 or 5 times during the growing season. The southern rust was always more severe on the early planted corn compared to the late planted corn, right? Even though they both got inoculum at the same time, right? The late planted would have just been a growth stage or 2 behind the early planted. But by the end of the growing season, the late planted had more severe disease than the early planted, because obviously the early planted matured, right?
Yeah.
There's no green leaves left, and so the southern rust can't infect it. So we didn't get another cycle of southern rust infection onto the early planted, but we did get a couple more cycles onto the late planted. And so we ended up with more severe disease on the late planted.
Have you been able to assess what you think the yield impact of that is?
Not yet, still waiting for yields.
And I would say with that too, we traditionally talk about later planted corn being more, right? In
Yes.
general, getting,
Yep,
getting
we
more
always
disease.
say that.
Yeah,
Yep.
it is more probably higher risk to get disease, but as the disease comes in, I think there is something about the leaf age. We are seeing more disease developing on the older leaves. And, so on August 2nd, the early planted had more, but by the time September 2nd came,
And yet, when we say older, that's not distinguishing between vegetative and reproductive with southern rust specifically, right?
No,
Just
not,
older,
yeah.
older leaves, whether it's vegetative or
Yeah.
reproductive stages. Yeah.
Every
So-
time we do these, I almost laugh because, to your point, it's so endlessly complex, and we want there to be an answer so bad. I mean, we just
Yeah, yeah.
want to call and have one of you say, "Do this on this date, and it will solve your problems." And it's never
Well-
never quite that simple. But
One of the questions that I've had, and I don't know if Daren wants me to bring this up, but I was wondering whether the corn kind of sees the finish line, right? And it's like, okay, I see the finish line. I'm just filling my grain. I don't care. I'm not defending myself against anything anymore. And I'm just going to fill my grain. And so one of the questions that I've had is, if we did are there any gene expression studies that have been done, right, where we could look at defense? What I'm hypothesizing is that defense gene expression goes down the more mature the corn gets.
Yeah.
And so I
And
did,
molecular training and research you're doing is really paying off now, huh? Thinking about.
and so, yeah. And so, I mean, I did reach out to a couple of people who I know do some work in that sort of area, but they didn't know of any work that has been done along those lines. So
That's pretty cool. Yeah.
Yeah.
The difference between like your PAMPs and MAMPs, some of that stuff,
Yes.
at R1 versus R3 versus R4,
exactly.
or even V18, V16,
Yeah,
right?
yeah.
I do know that that's one of the challenges when we're talking just at a commercial level to growers is, we see if you're on the field a lot in the middle of the fungicide season and when those AIs are active, if you spend enough time out there, you can see the control. You can see when it starts to break. You can see when the disease was there and it's applied where,
Yeah.
where it's having an impact. But if you go right in a combine today, even the fields that were sprayed with fungicide, a lot of them are just completely riddled with the disease. And so then it's counterintuitive if you're the producer, because you're like,
Yeah.
hey, didn't I spray a control on this? But
Yeah,
but
if you're not tracking it throughout the
Yeah.
Yeah.
because you
absolutely.
can have a treated field and an untreated field that don't look all that different by the time you get in the combine, but
Yep.
the timing was so much different. So, a couple more questions about Southern, and then we'll move on. So when we have that spore movement comes into the state, we have the disease triangle. So the inoculum lands. How long do I have before I can see that lesion expression?
Under optimal conditions, about 5 to 7 days.
That
So
quick, huh?
it's very quick. If compared to other diseases like gray leaf spot, where you're looking at 10 to 14 days, I think northern corn leaf blight is 7 to 10, and tar spot, I want to say, is like 14 to 21 days. So yeah, southern rust is the disease cycle is very, very quick.
So then once we get a spore that lands, germinates, infects a leaf, 5 to 7 days, you can have a lesion show up.
Yep.
How
With
long?
more spores.
Yeah, I was gonna say that's what I was getting to. So if we have environmental conditions, which this year were favorable the entire year, how long until that lesion begins producing spores and those spores
That's
reinfect?
7 days.
5 to
Yeah,
7
5
days.
to 7 days.
Yeah.
Yeah.
Man,
So that means we're getting new, every week. So we're going from one spore giving us one lesion
yeah.
to a week later giving us, I don't know,
Well, that was my other question.
100 spores.
you
Those
talk-
spores landing on
We're going
giving
to
us
use plant pathology math. It goes everyone produces 100 spores. It takes exactly 10
And,
days.
I mean, that's part of the conversation I have with growers. We look at some of these diseases like tar spot in southern rust. I mean, they just produce so many spores compared to some of the other diseases
Yeah.
we're used to. do we have an idea how many spores? I mean, I remember hearing some data on spore production from tar spot, tar spot lesion. Have we ever looked at spores produced in a southern rust lesion? Because the way it blows up, it's got to just be immense.
Yeah. I know that for northern corn leaf blight, there's like 500,000 spores produced per square inch of lesion.
That is also a lot.
Yeah, and the weed scientists...
It's much more than 100.
Yeah.
Yeah. The weed scientists think they have something with us, right, when they talk about the number of seeds that water hemp produces.
Yeah.
And I'm like, you ain't got nothing on a fungus.
That's hilarious. Yeah.
This is her talking trash. Right
Yeah, we'll send it over.
yeah.
Man, that's a lot. No, I mean that kind of leads into covering the science portion with part one. We got a lot of different diseases to talk about. I'm sure we'll come back to southern rust with stuff that pops in our head. But what's your thoughts on tar spot in 2025, and more importantly, thinking about, it's probably been a year and a half, maybe even 2 years since we've had Darcy on and just talked about tar spot. I feel like was as much research as we were doing. Have we learned anything more on dormancy, water, humidity requirements, moisture requirements that maybe was a little bit different than a year and a half ago, or just what's our newest thoughts on the science behind tar spot?
Well, I have one observation, and then I'll let her talk about the science. But I would say that the one thing that was interesting for me was tar spot was found a little bit earlier than southern rust this year, and we actually got quite a bit of criticism because we weren't talking about tar spot as much as some of the companies wanted us to be, because we started looking at the southern rust as more of a problem as soon as with the conditions, and our predictive models were showing that tar spot risk was pretty low and
Zero,
and it was really driven by temperature, and then as that played out, I mean, I saw one field in southwest Iowa that had quite a bit of tar spot by, end of August or, third week of August. But there wasn't really a lot of tar spot out there this year. But, it got here earlier, and it had the same conditions that southern rust had. So, and it produces spore. It knows how to take over a crop pretty easily. So, I think it's just a great example of we do need to be paying attention to that third angle on the triangle of the environmental conditions and how that may affect sort of the management decisions that we're making, and we'll talk management in the second half. But I think just that example is so stark this year, where we had inoculum, we have the corn hybrids, we have everything's out there.
We were just waiting for it to blow up because it was there so early.
Right, and then and one disease won, and what the difference is like just the preference in temperature ultimately.
So can I ask a question about that? And Allison, maybe if you could give me guidance on this. So when we think about the disease triangle, was it as simple as the scale was tipped slightly in southern rust's favor? And then I heard you say something in the beginning of the show that you think we literally just lost the real estate battle. I mean, tar spot lost the surface area battle to southern rust. Is it really that simple? Because I remember talking to Darcy a couple of years ago, and I mean, it sounded like if tar spot was here, it took 24 hours of leaf wetness, and it was gonna, just devastate our crops. Well, that certainly doesn't seem like what has really manifested itself the last couple of years. We're finding it relatively early, and we certainly had leaf wetness. We had a ton of leaf wetness. So maybe give me your insight on why wasn't tar spot worse?
too warm, way too warm for tar spot. Okay. So tar spot prefers much cooler conditions, like 68 to 75. So, and I point a finger at temperature. We had the right temperature for southern rust. We had completely the wrong temperature for tar spot. And then by the time we cooled down, we had that cool week, third week of August, was it, right? Mm-hmm. We cooled down, but then there was no real estate, right? Because the southern rust was there and it had just taken over all the leaf tissue. But temperature definitely, and if you look on the Crop Protection Network site at the prediction models for tar spot, I mean, we were at 100% the middle of June, and by the middle of July, we were down at 0. From the middle of July all the way through to middle of August, it was just 0 risk.
Do we know what the impact of a 24-hour average temperature is? You know, you could have warm days, cool nights. What's the impact there? Because I'm still
Good
thinking.
question.
I'm still thinking back to last year where we just had like 3 weeks or a month of just perfect conditions for what we thought was tar spot. Remember,
Yeah.
it was dewy, cloudy in the morning. We were getting rain every few days, and you would just see a cloud cover, and we were just expecting it to flare up, and it never did.
So I think according to Damon's model, Damon Smith from University of Wisconsin, we need to have 30 days where the temperature is, I don't know, around about 68 to 70. I can't actually remember.
So like for the average temperature
Yes.
in a 24-hour period.
period, yeah. And we need 30 days of that. So you can see why we get tar spot early on in the growing season, right?
Mhm.
It's no surprise to find tar spot in June because the corn's been out there for 6-8 weeks. It's been under the perfect conditions for tar spot to develop. But then once we get into July and August, we get too warm, and then we'll cool down. Okay, so but I still have some questions there because even though we get warm, by the time we get into August, then all of a sudden we have this sudden flush, right, of tar spot.
What's
But
it doing
we
that whole?
haven't- But we haven't had the 30 days of cool conditions.
Yeah.
So
Well, I remember you and I having those conversations. I mean, it seemed like last year, almost daily, we were on the phone, and at 10 in the morning, it was overcast, the air was wet, but then by 3 or 4 in the afternoon, it'd be 90, and that cloud cover would blow off, and so it always just made me wonder: is it just sitting right there, and it's so close, and then we come in and bake it off, and then repeat that cycle or whatever?
Is it just
but-
hanging out in the leaf, waiting for that month and a half, 2-month period?
yeah.
I mean, not waiting, but it's
Right.
there, and then the second we start having those
Yeah.
conditions, it just flares up.
You're referencing, that where we were a couple years ago to where we are now. We're starting to hone in maybe our understanding of the triggers for these diseases. But then you brought up skepticism around predictive modeling. What makes it? Sorry, this is not even close to any of what you wrote. But what makes it so difficult then? If we kind of know most of those triggers, what makes it so difficult that I can't just get an alert on my phone that says, "Hey, you just hit the 28th day of these temperatures, you need to be, have your head on a swivel for tar spot or southern rust or whatever"? Why is that as complicated?
So first of all of our prediction models rely on weather, right? And how accurate are we predicting the weather?
We're going backwards. I
10,
feel like.
yeah.
Yeah, rubbish.
So
Rubbish.
Yeah, we are rubbish at predicting the weather.
so therein lies the problem, right?
Yeah.
And so, and that was one of the things that I talked about this year, is that those prediction models are very valuable, and I feel like we're getting better at them, but I think sometimes people get frustrated because they don't work like they want them to. But how many people look at their weather app every single day? There you go, Sean,
absolutely.
right? And that weather app is, you just told me, rubbish,
Yeah.
right?
Yeah.
So,
I would say to that, to play both sides, we are definitely getting worse, I feel like, with predicting the weather that current day versus 2 weeks. But at the same time, we should be able to look at weather stations and accurately track the weather from the past 2 weeks.
yes.
You know what I mean?
Yeah.
That should be as accurate as, you know what I mean?
But you could certainly say maybe we're getting worse, and this is not to start a political conversation. Maybe we're getting worse, but I also think the weather is just incredibly variable, right?
Yeah.
I mean,
Yes.
you look at 2 years of extreme drought, perfectly timed rains, and then this year, I think the day we started combining, we were at 175% of our annual total rainfall.
Yeah.
So
Yep.
it's, I don't. The day-to-day variance is high, but so is the annual variance, and so it does seem like it'd be a really difficult job. But
Yeah.
it seems to me like, I know, like when we sit and we think about an advisor role, and you guys are in that too, but it's just frustrating that it's not easier to say, okay, there was the
Yeah.
trigger.
Oh,
now
yeah.
now we know now, and I feel like we kind of know that, and we're collectively pretty good at that, like with social media and how quickly information travels. It's not like anyone was caught off guard, once southern rust started hitting, but certainly it wasn't like we were getting all these like
Well, if
warnings
someone
coming.
had told me that it was going to rain for the next 6 to 8 weeks, every
Yeah.
3 or 4
Yeah.
days, and the temperatures were going to be in the mid 70s you know.
I think
I
more
would have
growers would
said,
have put second
get
half on.
out there and spray, spray,
Yeah.
spray.
But you would have rolled your eyes and went, yeah, but the weather forecast is probably going to be wrong. So yeah, let's wait and see. I mean, that's what we do.
You
but
play the odds, it's going to dry
Right.
out at some point, right?
Yeah.
Andrew, keep us moving.
Yeah, no, let's continue to build on you know, we got some additional information on the science behind tar spot, maybe why it's flared up, why it hasn't. Let's talk about this new fun one that.
Can I actually? Sorry, I just want one more scientific question
You
about
can.
tar spot. I apologize for interrupting you.
Approved.
Okay. Do we have any, thinking back to maybe the guidance from 2 years ago, and the evolution of our understanding, do we have any scientific, not management, but any scientific updates? Is there anything that we really feel like, okay, we understand, the temperature ranges, we're getting to understand those a little bit more. Maybe we have a more narrow understanding. Is there anything else that scientifically we understand at a different level than 18 months ago?
I'm sure there probably is, but I can't think of it at the moment. Darren, can you think
You
of
have us on the
anything?
spot here.
Yeah. I'm just like, Darcy and
See,
Marty are going to be...
all of our listeners here, I set
Right.
you
Yeah,
guys
yeah.
up, and now.
Yeah.
Right.
And our Darcy, Marty, Damon, they're going to be rolling their eyes out there thinking, what? You didn't read our paper?
We'll get them to chime in. They can.
Yeah.
They can.
Yeah,
we don't want to
we
steal their thunder for
Yes,
future episodes.
exactly. Yeah, we're not stealing. Yeah,
That was a
yeah.
perfect comeback right there. Let's talk about this new one. And man, we don't have to dig deep because we just had a really good episode with Dr. Maida Duffick from Oklahoma State talking about this new guy in town, corn stunt. And man, talk about a tricky disease. You know, you got 3 different organisms here in the U.S., 4 and globally. You know, where it's typically associated with. Just give us your thoughts on that. You know, from the science perspective, we'll talk management obviously with the insect here in part 2, but from a science perspective with such a weird disease that has multiple organisms associated, just give us your thoughts on that one.
Yeah, I'm glad that Allison's the corn pathologist. Is really my conclusion. No, it is it's always interesting when you get something as complicated as it is, because there's also an interaction with the leafhopper and then like sort of the learning curve as to what leafhopper and like when and the timing and like what is it carrying, and so it is like with southern rust and tar spot, like, you know, you have one organism and sort of it's already complicated. You have different hybrids, you have different decisions, but with corn stunt it adds an extra layer, and so you're looking at you know, it's been a problem 50, 70 years ago, and then it's sort of now it's sort of, and then
Again,
it sort
why
of
now?
went to other continents and hung out there for a little bit, and now, you know, it's like, okay, now
world
we're going to
tour.
come back. Yeah, we're going to come back and like randomly like start, you know, sort of making a fuss here back in the U.S. So we have a lot to learn. Is it you know, we just introduced some really susceptible hybrids, and we sort of got to flush those out and reset, or is it, okay, new reality, then leafhopper's more efficient, and now it's doing this, and so a lot to learn. I'm glad we have young scientists that are diving in head first on with this one, and we sort of are there to help them. I know that we have plots that we go out and sample, and then we ship all of the stuff over to their labs and let them do the testing.
Did either of you see it this year? Because I
No,
didn't even know.
so-
I didn't even know it was confirmed in Iowa last year when we posted the episode. I did not know that it was confirmed in Iowa last year.
I don't. So, yeah, I listened to the episode, and so I noticed that Iowa wasn't mentioned, and that's because we have a virologist here who had all the samples, and so I'm not 100% sure of whether he found corn stunt. I think he did detect it.
Yeah.
here.
Yeah, he did.
Yeah. But what I was going to say was, yeah, so it's transmitted by the corn leafhopper, and then it's the 3 pathogens, but you don't need all 3 to get the disease. You can have 1, 2, or all 3, right? The other thing, so what we've been doing this year is, as Daren mentioned, we've had sticky traps out at plots, or 4 locations in Iowa. So southwest, northwest, central Iowa, and then southeast. We have yet to detect the corn leafhopper here in Iowa this growing season. So if we haven't had the vector, then we can't get the disease. And at the moment, that's the only vector that we know of, right, for corn stunt.
It's been confirmed in Iowa yet this year, hasn't it? I thought.
The leafhopper?
No, the disease.
No.
Oh, I thought it had been.
No, I have not
Oh, this is
heard
gonna
that
be
it's
fun.
been.
We're going to fact check.
I know.
yeah.
We're going to fact check you guys.
Everything needs to be fact checked.
Yeah.
Strong
Not
no.
that- So I think that, I think we have to be careful, right? Because you can get red plants when
Yeah.
there's no ear,
Yeah.
right? And often those plants at the edge of a field, right, where a deer's come along or a raccoon or whatever and chomped off the ear
Yeah,
or whatever.
yeah.
So just because you see red plants doesn't mean that you've got corn stunt,
Yeah.
right?
And we have herbicides that will stunt a plant.
Yeah.
And yeah.
Exactly. And so, and then I think the other thing about corn stunt that we need to remember is that when the corn leafhopper picks up the pathogens, right, that pathogen has to go through its body, and that takes, 21 days, I think.
Yeah, I
my
remember
era
27
said?
or 20, yeah,
21,
21,
30
27,
days.
something like that.
Then they've got to infect the plant. Then it takes another 21 days until we see the symptoms, right? Where you get huge yield losses from corn stunt is when you get that infection happening V6, V12, when that ear is being set, right? And it looks like from this year that the corn leafhopper did not overwinter here in Iowa. So that means it has to come up from the south. So if it's really going to cause a big issue, it needs to get here.
A lot earlier.
It apparently
In
doesn't
the
move as
middle of
good as
June.
southern rust
Right.
spores.
Yes.
It's
Needs to
not in
take
the
a
same
ride with the
wind.
southern rust
Yeah.
spores,
Yeah,
and it'd
yeah.
be a lot better. So.
So yeah, I mean, it's got to get up here mid-May to mid-June, right, to cause any really serious damage. And so, yeah, I mean, I've enjoyed monitoring for the leafhopper. We've been working with Aaron Hodgson on that. And, yeah, I guess I'm not as concerned about it as maybe some other people are. And maybe I'll eat my hat in a couple of years and say, oh, crud.
I bet there was someone 20 years ago that said the same thing about southern rust.
Yeah, exactly.
Hopefully they're on tape or something.
Yeah.
recorded.
No.
Yeah.
But, yeah, those are, so,
Yeah, that's a tricky one. Time will tell. We'll have
Yes,
to check. 'Cause
exactly.
Like I said, I thought I heard it was confirmed in Iowa, but I don't know for sure either. And you guys would know more than I would, so apparently it must
Yeah.
not have been.
the only person who can confirm it would be Alejandro.
Mm-hmm.
Because, yeah, unless somebody sent samples down to my era. But,
Yeah.
yeah.
Cool. That's definitely one to keep an eye on. Let's talk another new one. Man, a flush of these new diseases.
Listen, no one's
this-
going to want to listen to
this-
this. If
Job
everybody's
security.
not having enough fun right now.
So
everyone's
this
gonna be like,
was
"I'm out."
confirmed in Iowa, curvularia,
Yeah.
right? And
Yeah.
I'll never forget. I remember. I always like to say it's eye spot on steroids. I think that's a good description of it. I've heard other people use that. I remember, boy, it was 2017. I was in a field and I was taking notes on disease at the time, and I remember finding something. I had no clue what it was at the time, but it looked just like that. And I remember taking it to Ed, and we ran samples. I don't remember what it was. No one knew what it was. I remember showed you, and it looked just like that. Again, I doubt it was curvularia, but it was probably some genetic response. It was through the entire plot. Give me your insight on curvularia. What do we know about the science? I mean, this is something that they've dealt with in the southeast, warmer climates. It's found its way up to the Corn Belt. Do we know if it overwinters? Some of the science behind this pathogen.
So I suspect it overwinters, but I have a master's student who will be doing an overwintering study this year. So we're collaborating with our colleagues in the Corn Belt, right, who have curvularia. So right now I hope they're collecting samples, and we'll be leaving those out over the winter, and then Luxon will be looking at those in spring to see whether they're still viable.
Okay.
But
So you're going to take curvularia infected residue.
Mm-hmm.
Either
Leave it.
Either it's out there or put it back out there to see if.
We're going to put it back out there in residue bags. So
Yeah.
we'll have it on the surface, and then we'll have it buried 3 to 4 inches below the surface.
Okay.
And then,
See if it survives the winter.
yeah. And I want those bags out by mid-October, right?
Mhm.
So that it has that end of fall. And then we'll get the bags April, May, just before planting, and just look for, do the spores germinate? Can they grow on a petri dish? And we might try and do some infection studies. So, yeah.
Where was that confirmed in Iowa? Like, where did you see it?
Oh, my goodness.
Or was
Central
it-
Iowa, Northeast Iowa,
I'm not going to tell the
Southeast
story of
Iowa.
my.
Tell the story.
No, this shows how stupid I am, maybe, but which you guys will enjoy. But I was teaching 279, and so Andrew used to teach us, and so it's a course that we bring the students in 2 weeks before classes start, and they sort of get out into the field and they look at things, and so they get tortured with about a 3-hour session on plant diseases. Sit in the the classroom for the first hour. We sort of go through diseases and sort of the update, and then we go out to the field and have them go out and look for things, and then we bring them all back, and then they try to identify them, and then we sort of talk about it and sort of
Games that you would have challenged me on initially early in my career and
So-
cheated on against, stacked the deck. we'll
you
say.
never beat me. That's a whole different episode, though. So anyway, most of the students went out and they all found southern rust or sudden death syndrome on soybean, and it's pretty easy. And like one student came back and he brought back what I said was eye spot, and we brought it back, and I was like, oh, this is an
This was
interesting
at the
one.
field lab?
At the field lab, so right here in Boone, or just outside of Boone. And so I send the leaf around. I was like, this is an interesting one, guys. I said, hold it up to the light. Sort of I'm teaching them, like, hey, the sun, backlit leaves, and you can see all kinds of extra details. And I said, whenever you see something like this, I said, it really comes down to it's going to be eye spot or curvularia. I said, now it's context. I was like, where are you standing? We're standing here in Iowa. So it's not going to be curvularia because that's a southern disease. And so this is going to be eye spot. So good find. I said, it's a little bit warm for eye spot, but, hey, good find. That's really good. And then an hour later, she texts me and she's like, hey, just confirmed curvularia leaf spot here in Iowa. I was like, oh, crap. I was like, I never went back and told the students that they found I was wrong. Or anything.
Hopefully
like,
none of them are listening.
So if they were listening, like whoever it was that ripped off that leaf, they probably had the first find of a curvularia leaf spot, or maybe yours was first. But
You know what? If I beat you, it was by like 30 seconds.
Yeah, it's
and we were literally half a mile apart.
yeah.
That's amazing.
Yeah.
So
it
you
was
kind of led
funny.
into it. So I mean that kind of separates curvularia and eye spot, just temperature preferences.
Oh,
so,
yeah.
so
Yeah.
curvularia
Definitely.
likes a little bit warmer.
Yeah, much warmer. Yeah.
Okay.
Yeah.
Okay. That's good to know. Yeah, that's going to be a tricky one, but I know actually a throwback too, just to reference Dr. Travis Faskey's episode from Arkansas. That was really good. If anybody wants information more in depth talking about southern rust, that was a good one to go back and listen to.
Yeah,
I-
we're
I
going to
know-
need to tag a few of these because I think there's some really good. I mean,
Yeah.
obviously, it's a pretty small network of people that we've spent time with. So it'd be fun to tag some of those people in this.
Yeah, well, curvularia, I mean, that's one that it sounds like we can easily manage with a fungicide, but again, that thing can flare up pretty quickly. I mean,
The thing is it's going to flare up probably more in the upper canopy, and it's going to sort of take on the leaves that we maybe protected with the fungicide, and it's just
That's
it's just
definitely what
add
I
it.
saw.
Right, added on to tar spot and southern rust and everything else. And it alone probably isn't going to be a huge yield loss issue, but combine it with some of the other stuff, and then it's just add one more thing, consideration.
I'm looking forward to the management discussion because now we got in southern rust, you never know what it's going to be like, but now we got diseases that whether it's cool or warm,
Exactly.
just are going to
Yeah.
thrive and multiply like no other, right?
Yeah.
Yeah.
Well, I think the days of, we just have to be. Just feels like we have to think almost entirely different than we've ever thought about the use of these tools and timing, and
Yeah.
a lot more in tune with what's going on with Mother Nature. But let's keep hammering away here. This is probably one that I feel like we certainly had pretty significant impact on which is crown rot, for a lot of the reasons that we've already talked about. But I guess let's dive into that conversation.
It's good too, because I mean we got one of the experts, Allison, and one of my favorite people on planet Earth, Dr. Tamara Jackson Zims from Nebraska, have done a lot of cool research looking at PAM and some of the fusarium species associated with crown rot, or what we think is associated with crown rot. So kind of build on that. I mean, just for a lot of listeners, PAM would probably be kind of new, and I'll put you on the spot. What does PAM stand for, and tell me about it.
So PAM, it stands for Phytocytroma ambiguum.
There we go.
which is
I
would've-
swear they all make up words.
That's a
just to
made-up word, yeah.
I don't. Yeah, that's just to make sure that we
I
don't.
think it's something you spray on your pan. to
exactly.
Make
Yeah,
sure like
yeah.
the
Make sure my eggs don't stick.
eggs don't stick. Yeah.
crown rot. So we started working on PAM because when we had crown rot plants coming into the lab, we would get the usual fusarium species, but a lot of times we would also keep isolating PAM. And so when we went and checked in the literature, PAM had been reported as causing crown rot, root rot on corn in other parts of the world. So Brazil, South Africa, Europe. And so Daniel, my student from Kenya, that's what he's been working on. So we've taken isolates that we got from crown rot samples collected by ourselves and by Bayer agronomists. And we've done pathogenicity tests to see whether we can cause disease on corn. That was a challenge, but we have managed to show that we can cause crown rot and root rot on the corn. We've been doing fungicide sensitivity assays just to try and understand if there's a seed treatment fungicide maybe or an in-furrow fungicide that could help. Although I have to admit that I'm not sure that infection happens at planting.
Hey, perfect. We're building that.
Yes.
That was going to be the next question.
I know.
we had a lot of assumptions based on kind of what we think happens.
Mm-hmm.
Infects early, maybe lives as an endophyte, and then there's some trigger that causes that organism to become pathogenic.
Yeah.
Has your research in the last 2 to 3 years changed maybe some of your views on infection timing?
Yeah, and I'm not sure that it's really based on the research, but just maybe thinking about it a little bit more. In our field trials that we did for crown rot, so we have, 9, 10 site years of data where we've tested different in-furrow fungicides, seed treatments, foliar fungicides, to see if we can manage crown rot. The problem in all of those site years is that we never got crown rot. So in the control, right? So there's no crown rot in the control, so we can't tell if any of our treatments worked. So yeah, that's what's had me thinking about infection. So one of the studies that we've been doing the last couple of years, unfortunately we don't have crown rot, but I started to think about, if you think about a corn seedling, you have the seed and the radicle and the seminal roots, and then you have the mesocotyl, right? And then you have the crown. So if we're getting infection through the seedling, that pathogen has to get all the way up through the mesocotyl and into the crown. And I'm not sure that it can do that. It's a pretty slow-growing organism in the lab. So and we know that if we take a toothpick with PAM and stick it into the crown, we can get crown rot, right? So one of the questions that we've had is maybe there's an injury to those nodal roots which are coming out of the crown, right? And that's how PAM is getting in and causing infection in that crown. So we did a couple of studies and where we went in and we injured one of the nodal roots, and then we actually inoculated those nodal roots to see if we get crown rot. We didn't get crown rot. So I don't know. But, one of the ideas was that maybe those rootworms injuring those nodal roots is allowing that fusarium. This past year, we actually did fusarium graminearum and we did PAM, and we inoculated, and we didn't get any crown rot in that field. And that field is the field that you and I visited when you were still working on your PhD, I think, and we were doing physoderma work there. And it was the first time we saw tar spot, and there was and there was also tons of crown rot in that field. So I know that it's a really good field for crown rot. But yeah, I think
it continues to be challenging, because I know I had some assumptions and ideas, similar to your crown rot thing. I had some ideas just looking at root growth, some of the things maybe we'll look at. Does stagnant root growth open that window for organisms to enter? But I know this year is really throwing a wrench in some of my thoughts, because products that typically get crown rot haven't got it this year, and vice versa. Products that typically don't get crown rot have got it this year. No rhyme or reason. Don't know. It's just the second I feel like we're starting to get closer to an answer every year like this, it just throws a wrench in
Yeah.
kind of all
I mean,
my train of thoughts.
The best part about this conversation, though, for me is like you guys are experiencing the scientific process. Like you're trying something, it's not working, but you're continuing to make observations and then test different. I think like if we can get all of your listeners or like all of America, just not to make it political, but like just to appreciate like this conversation, like it's such a beautiful thing. It's just like we don't know what we're doing sometimes.
Yeah.
Like that's okay to say that, and then they come back and be like, okay, well, like it's great. Like you're just making observations. Like, well, what about this? What about that? Hit it from this angle. Okay, this didn't work. Okay, go back to the drawing board.
Yep.
And it's going to be based on conversations like, hey, this didn't make sense out in the field, and you're bringing that information over, and it's cool.
I'm going to dive in there for 1 second because that's one of the things that I would say has probably, like, if there's been a light bulb for me or just kind of the experience of doing this podcast with Andrew is, I don't want to say like we're lazy as farmers, but really just figure out the solution and charge me for it, and I'll be happy to buy it from you, right? And I think that there's just a massive opportunity to appreciate and participate from a commercial producer standpoint because, I'll be honest, I had so little understanding of how important the crop protection network is and the land grant universities and how tied in it is to the major manufacturers and how critical the work you guys are doing helps them understand things and vice versa. There's just been, for me, it's been really eye-opening. I do hope our listeners understand that. And I think we've got a lot of brilliant listeners that probably have a ton of ideas, and they probably don't even know to pick the phone up and call and say, hey, I made this observation. It's amazing how often we aren't the smartest guys in the room when we're having conversations with these growers because they're the ones that are literally doing it every single day and at volumes that, far exceed any of what we get to do. And so I do really appreciate that call. And it's one of my main hopes is that listeners would understand how just the humbling efforts that go into trying to understand these incredibly complex things, So
Well said.
And that's one of the things that I said at the beginning, right? But that's the cool thing about being an extension is interacting with the ag business, interacting with farmers, because they come up with ideas and all observations, right? And you're like, and why did this happen? And I'm like, oh, that's great. Let me think about this, right?
Yeah, absolutely.
So, yeah.
Well, we better wrap this science portion up. I wanted to touch on some soybean diseases.
Soybeans?
Again, what a weird year. You got certain diseases that you think would probably, and SDS did show up bad, and we'll talk a little bit about that, the science. But frog eye leaf spot, some of these other diseases, they were there, but I don't know if they were maybe there at what we thought they would be. Let's talk about that. We'll go SDS first. What do you see, and when we'll start? When's the infection take place?
Yeah. SDS was by far the most prevalent one that we had, and we're sitting here in Iowa, and so I would say if you go to any of the neighboring states of Iowa, they would have red crown rot on their mind.
Yeah.
And that conversation.
Every time I hear that, I'm like, Daren's trying to find it in Iowa so we can confirm it, but I don't think he has yet.
I have tried so hard. we have spent a lot of hours driving around Iowa looking at
everything
patches.
That's red comes up fusarium,
right?
It?
Yeah, it hasn't happened yet, but we're still trying. Anyway, red crown rot is driving a conversation in Illinois, Indiana. It's a huge problem.
Yeah.
And but like here in Iowa this year, we had a lot of SDS, and it's I think we sort of alluded to it a little bit on in the corn side of things. Is like some people got out and planted really early this year, and so that, and then we had with the same rains that drove southern rust and some of the diseases in corn. Those are we used to call them SDS rains. You get a good SDS rain in the middle of summer, that's going to sort of drive it, and we had that for 4 weeks straight. So we were sort of anticipating, SDS showing up and with the early planting and all of that rain. So, and it didn't disappoint.
Yeah. So that's the one thing I wanted to ask. Do we know now, I know you've done a lot of research looking at SDS, it's kind of your area of expertise. Do we know now, is it a combination of early plant, we'll say wet soils and cool soils, or is it just wet soils anytime throughout the year can cause that infection and then later cause the toxin to move up?
Well, yeah, so I think you have to unpack it a little bit. So there's two phases to the disease. There's root rot and then the foliar stages. I think if you're just talking root rot, cool wet is probably more problematic. If you're talking the foliar stages, I think it's just wet.
So we can get symptomology, but we have to have the root rot. We can't get foliar symptoms without the root rot, correct? Or we traditionally thought. Do you think?
Stay tuned. I mean, this is an area that we're looking at. Like, we know. So I'll just describe one experiment that we did, and like I don't know where it's going, and like this is like you're sort of we're peeling back a little bit of science here, but I can you can take this mug right here, and I could grow the SDS pathogen on in a broth, and then what I can do is I can filter out all of the fungal parts of that, and what's left is called a culture filtrate. I can put that culture filtrate, which is just sort of the remnants or the toxins essentially of that's being produced by that fungus, in this cup, and I can take a completely healthy plant with the roots and stick it in, and 3 days later I'll have SDS symptoms.
Really?
No infection.
Great.
So so we know the SDS toxin can move through the roots, completely healthy roots. They don't need to be infected, and cause the foliar symptoms. Now, does that happen in the real world? Does that happen so? So I have a postdoc that's sort of trying to tease all of that apart and trying to figure out what is the importance. My guess right now would be, if you have root rot, it sort of gives you have easy access to the toxin. If you don't have root rot, then you probably have to have a little bit more extreme conditions for that toxin to get from the soil up to the top of the plant. But that's
interesting.
So that's an entire PhD
it's weird
thesis.
How it is. I mean,
right,
you learn this stuff. You guys have. I know. I learned it from you. You got the root rot. You
right.
can't. But things change, right?
So
that's fascinating.
And again, it could be completely wrong. Like so,
Yeah.
we could come back in a year or two and we'll be like, oh yeah, that. But like that's what we're sort of trying to peel apart right now. What's the importance of the root rot? What's the importance of the toxin that's in the soil that's not associated with the fungus that's on the roots?
So you could have the FV, the species that causes it, releasing
breaking
toxins
down
in the soil.
residue. Yeah. Just doing its job as a saprophyte in the soil, and then producing the toxin, and you get enough of that moisture.
It just moves with the water, follows.
Yeah. It could be hypothetically.
Yeah.
So that's what we're testing.
That's terrifying.
Terrifying. Waiting for that.
I feel like anytime we do scientific updates, it's like we never solve anything. We just add more and more potential terrifying things to the future. It's kind of what we do. But guys, for the sake of time, I guess I'll give you just a generic question, and then we'll wrap up science, because I know we have a lot to talk about in the management side of this. As you think through kind of the whole disease spectrum and what you guys are studying, is there anything that we haven't talked about that you want our listeners to be aware of, specifically thinking scientific revelation in regards to disease pathogens, that sort of thing? If not, we'll hit some management.
I'll start. I mean, we didn't talk about this very specifically, and I think it's a quick story, but I did say that we went out and we were trying to find septoria brown spot to specifically look at fungicide resistance. So sort of, and what we were finding was actually a lot of leaves that were infected with Choletotricum or anthracnose, and so I think it's always a good reminder. And you're saying, you're skeptical about like being able to just take photos and say or like AI to identify some of these things. I think plant pathologists have a hard time identifying some of these things, and we need to always continually go back. Like my silly story about curvularia and eye spot. Like we get it wrong.
We're all
too.
still learning.
Right.
Yeah.
And so and it is complicated, and it's not easy to identify these things. There's a lot of lookalikes. There's a lot of things that are going on. Red crown rot looks just like SDS. Like everyone that just says, you see a yellow patch, that's 90% chance that it's SDS. Well, you can't do that in Illinois anymore. Like, you can't do that in Indiana. So. We have a lot of. It's good to continually go back and revisit some of these questions and look at these things and try to figure out why the heck am I seeing anthracnose leaf blight the last 2 years, which I've never even considered it. It's always been septoria brown spot, and what's the importance of that? And so, put a student on it. We start looking at it. We start looking at what's going on and asking the questions why, and go from there.
Yeah, that's interesting. You think of Choletotricum, it's a corn disease, right?
Yeah. But we're finding it on soybeans.
And mangoes and avocados. causes anthracnose on a lot of
My
different
mango and
crops.
avocado tree in my farm died.
Just didn't survive the winter.
I'm going to skip all the jokes I'd like to make that I'd have to immediately edit out anyway. So anything to add?
I was going to say, thinking about the southern rust, and I think I mentioned about common rust with the adult plant maturity. But actually, and Kirsten Wise brought this up at one of our teleconferences that we had, that she thought that common rust was coming in on top of the southern rust towards the end of the growing season. And yes, we saw the same thing here in Iowa. So when we got to the end of the growing season, all of that rust on the leaves was not all southern rust. There was some common rust there as well.
I'm glad you reminded me of that. That was a question I wanted to address, and I forgot. As we progress towards the end of the growing season, southern rust kind of switches up its reproductive stage, and often it gets confused for tar spot. Yes. So tell me about that, the Telia spore, and just what that. I wouldn't say trigger, but why the change? What's going on there, and then why the difference in appearance? Because that's good. I mean, we don't want to mistake tar spot for southern rust, right?
So southern rust produces 5 types of spores, and we know the orange spores. Those are the ones that infect the corn leaf, and then we see the orange rust on the corn leaves, right? And then as we get to the end of the growing season, I'm not sure whether it's cooling off, the temperature cooling off, or the corn plant maturing, but the pathogen will now start to produce black spores, which are Telia spores. Those are more embedded in the tissue. They're not loose on the surface of the tissue like the orange spores, and so that's why they can be confused with tar spot. Tar spot is much blacker. They tend to be more dark brown. And then tar spot has those spindle type lesions, right? So you can tell those apart. And then the other thing that occurred to me the other day when I was thinking about southern rust and the teliospores cannot infect corn. So
you have no reinfection.
Yes.
Okay.
So as we think about southern rust, right, there's a lot of talk out there at the moment that southern rust can overwinter, right? It might be able to overwinter in Texas. Somebody told me, well, it overwinters in Texas. Yes, because Texas has green tissue in February, right? In Iowa, we don't have any green tissue. So first of all, we don't have the green tissue for it to overwinter on. But the other thing is that pathogen now is changing onto these Telia spores, right? So the orange spores are finished. They're dead, they're not going to be produced anymore, and then those teliospores are not going to infect. Even if they could survive in the soil, they can't infect corn.
That's good to know. Lots of questions and confusions out there, I think, with that, so it's good. Despite the horrible southern rust year, it's good we can at least clear that up.
Yeah, we start with a clean slate next year.
I just, every time we have these conversations, this stuff is so endlessly complex and then infinitely variable, right? And you've got people that have dedicated your entire lives to understanding it, and it's constantly evolving and changing. And so really looking forward to a conversation around management. That was hardly efficient, but I think very very full of
lots
great
of good
information.
info,
and yeah, I mean, you guys are just such a wonderful resource. I know on behalf of myself and Andrew and our listeners, we're just incredibly appreciative of you guys being willing to be so generous with your time and
be intimidated
freely give of your knowledge. So thank you guys, and look forward to coming back and talking about how to manage these complex situations.
Thank you.
Thanks for having us.
Thanks, guys.